Lab we're going to do is the importance of hand washing. It's a very important skill that's not practiced enough, especially in the healthcare field, and that's why we have tons and tons of nosocomial infections. Whereas this is a multi-part lab, and then this is probably the first time you're ever going to deal with petri dishes, so I'm going to show you about the petri dish. Petri dish contains a lid and a top and a bottom. The lid actually fits over the bottom, and you see you take it off. You need to leave these petri dishes closed as much as possible because microbes are present in the air. You can distinguish between the top and the bottom because, see, as you can see, the top fits over the bottom. The other thing is that as you look at the bottom, I'm just going to remove the top. You can see that little tan layer that's on the bottom. That's the agar. It contains a bunch of nutrients. And the type of agar we're going to be working with is called nutrient agar. It's a general purpose medium, and it allows us to grow a bunch of different types of microbes. I'm going to flip this plate over, and as you see on the bottom here, you can see that little cloudy rectangle right there. That's another way you can recognize the bottom. Okay. So that's the basics of a petri dish. Inside, the agar has a consistency of jello, so you have to be very, very careful when dealing with that because you can break it and you can puncture it. It becomes a pain in the butt to work with, as you will see. Whenever we label petri dishes, we always label the bottoms of the petri dishes. And in this lab, what you're going to be doing first is you're going to be doing a basic evaluation of yourself and seeing how well you wash your hands. Don't cheat on this. If you are one of those 2-second hand washers, just go ahead and do that. If you wash for a little bit longer, then that's fine. We want to see if what you're doing actually works. Okay, there's not going to be a grade for this. We just want you to actually see it. I know that whenever I go to the restroom, if guys are in there, sometimes, actually most of the time, they don't wash their hands whenever they leave. So think about that the next time you shake some guy's hand. But a lot of times, even when they do wash their hands, they recontaminate their hands even before they leave the restroom. They're doing a bunch of different things that we'll talk about in class. But you'll see some guys in there where they do the 2-second hand wash. So they just stick their hands in the water and then they go and they're done. If you're one of those, done that, then do it in this lab section right here. So when we label these, there will be Sharpies at your benches. They're usually in the top drawer. You can draw a line down the middle. One side you're going to put before, a B for before, other side A for after. Okay. You also put your initials and date on there. I don't know what date is, so initials and date will also be on there. So all this is in your lab. Okay? So you've labeled the bottom of that plate. Now after you've done that, what you're going to do is you're going to flip the plate over. Pay attention to which side is before and after. My before side is actually on the right side of the plate. Without talking, you're going to open the petri dish. You will take your fingers in one hand and you will gently press on the before side. Remember, this has a consistency of jello, so be very, very careful. If you got long fingernails, you need to put your fingers at a little bit of an angle. Okay, so you touch it 4 or 5 times, and then you close the lid. Then you're going to go to one of the sinks. There are sinks all around the room. There are 4 sinks on this side of the room, and there's one on this side of the room. Go to any one of the sinks, wash your hands as you normally would. Dry them with a paper towel and then come back. When you come back and your hands are dry, what you will do is you will open the petri dish. Once again, without talking, you're going to inoculate the after side. Same way, same hand. 4 or 5 times, pat it down, close it back up. What you're doing here when I say inoculate, that inoculation means to transfer microbes from one area to another. You are transferring microbes from your fingertips to this medium here, okay, and hopefully they'll be able to grow. Now, usually somewhere in the classroom there will be a pink tray. Right now the pink trays are way back there on the incubator, or the refrigerator. So I'm not going to go get it right now. What's very, very important is that whenever my phone going off. Whenever you put these, or whenever you're done with these plates, what you're going to do is you're going to put them on a pink tray. You're going to put them label side up. Okay, so it's going to be upside down. Your label's going to be on the top. That way you can see your name, and you can when you grab the plate. What you'll notice on some of the plates is that they have a lot of condensation on them. The condensation is going to be on the lid. Okay, until it's on the lid. If you look at it from the side, it's not on the bottom. Okay? No, it's not on the agar. So that isn't actually microbial growth. That's just water on the top that's there. It's not an issue as long as it doesn't fall on your microbes and stuff like that. So be very, very careful with that. So you can still use these plates. I get a lot of questions about whether or not you can use them. You can still use these plates. Just make sure you're careful with the water. So for most labs, whenever you are done inoculating the plates, they will be put on a pink tray, label side up. There are going to be a couple of exceptions we'll talk about a little bit later in some of the videos. Okay. So you're done with those plates. What I'll do is I'll incubate them at 37 degrees Celsius, which is body temperature, until the next class period. And what you're going to see is you're going to see little dots on each side, the before and the after on these plates. Those little dots are colonies, and each colony is just a bunch of microbes in one area. Each colony comes from a single cell that was transferred from your hand, your fingertips, to this plate. Okay, there'll be different sizes, shapes, colors, stuff like that. Won't be able to identify them based upon the colony color, shape, stuff like that, because there are a bunch of microbes that have the same exact colonies. They look the same when they grow as colonies. So hopefully you'll have a difference between the before and the after side. The next part of this lab, you're going to go to the sinks. And at each sink there will be one of these little glow germ kits. Inside the kit, there's glow germ powder, there's glow germ lotion, and there's a little light. In part 2 of this lab, what we're going to do is we're going to evaluate the 20-second rule. This is going to involve students 1, 2, and 3. You'll be getting in groups of at least 5 for this lab in this part. So students 1, 2, and 3 are going to be involved in part 2. And what we're going to do, as I said, we're going to evaluate the 20-second rule. These little lotions here, this little lotion here, will be placed on all 3 students' hands, students 1, 2, and 3. You're going to get it all over your hands. You're going to rub it back and forth. You're going to get it between your fingers on both hands. You're going to even get it in your cuticles by just rubbing your fingertips like this on both hands. Make sure you get it all the way up to the wrist area of the hands. Okay. Student 1 is going to get their hands contaminated. These aren't actual germs. It's just stuff that fluoresces under a black light. Okay. And that's all they're going to do. They're going to be the control. Okay. Student 2 is going to get their hands all filled with lotion, and they will wash their hands at the sink for 5 seconds. They need to be done in 5 seconds. So we're going to need a timekeeper to make sure that they stick to that. And it's going to be really, really fast. Okay, they're going to want to wash their hands for more time, but don't let them. Okay, so you need to wash your hands, and then once you wash your hands, you can dry your hands with a paper towel. Student 3 needs to wash your hands for 20 seconds. Okay, make sure that you follow the procedures in the lab. In the importance of hand washing, there's a little chart that I obtained from CDC, and it shows all the different steps for washing your hands. So make sure you do it correctly. You need warm water, you need soap during this procedure. So we're just going to evaluate. The only variable here is going to be time. It's going to be 5 versus 20 seconds. Okay. So that's part 2. Part 3 will be done at the same time. And part 3 involves the glow germ powder. Okay, make sure you use the appropriate item for each part. Part 2 involves the lotion. Part 3 involves the powder. And part 3 we're going to look at how much can be transferred in a simple handshake. How many germs can be transferred in a simple handshake? This is going to involve students 4 and 5. Student 4 will take this glow germ powder and they will put it all over their hands. So their hands are going to be white. All the way around, on the front and the back side of the hands. Very, very important to put on both sides. Okay. Once you do that, all you're going to do is you're going to introduce yourself to student 5 by shaking their hands. Say, hi, I am, and they're going to grab, student 5 is going to grab your hands and give a simple handshake. Okay. You take it away. After you've completed this part, both parts 2 and 3, you'll let me know. I will turn off the classroom lights, and that's when you get out your black light. Okay. With this light right here, it actually has 2 things. If you put the switch, you can see that switch there. Put the switch this way, it's a flashlight. If you put it this way, it's a black light. So I'm going to turn off the lights of the classroom, and you're going to turn on your black lights, and you're going to look at student 1, 2, 3, 4, and 5's hands. Okay. In part 2, you're looking to see how effective was 5 seconds versus 20 seconds. Where did the germs remain? Were there any germs left on Student three's hands after 20 seconds. Okay, you're going to evaluate all of that stuff to answer those questions. For part 3, how much can be transferred in a simple handshake? Do you really want to shake somebody's hands? How many times have you gone up to somebody and right before they shake your hand, they sneeze right into that hand or they cough right in that hand? And then you shake their hand anyway. Did you get sick after that? So these are important things that you have to think about, and we'll discuss these things in the classroom. So that's the importance of hand washing. When you are done with this lab, it's important that you turn off the black lights, put it in here along with the lotion and the powder nice and neat. I'm going to close these up. These remain by the sink. So for parts 2 and 3, you're going to be working at the sink. It'll be one group per sink. Okay, and try and make sure that you listen to me whenever I'm talking, because there are going to be a lot of people that are working during this part, and if you get involved with your own conversations, you're not going to be able to hear me, and I may give you actually some important information for the lab exam that you need for the lab exam during this time, and you may miss out on it. So just make sure you're paying attention during this part.